Hello, my name is Anna Toryńska-Sitarz. This short video covers the topic of engineering microbial hosts for the production of marine natural products. There are several ways to obtain natural products of marine origin. Among them we can distinguish cultivation and extraction, synthetic chemistry, and the method on which we will focus further in this video. Genetic engineering. Let's ask the expert why this method is useful in natural products discovery and production and how it works.
To obtain natural products from various organisms, we can use different methods. The first is, of course, the cultivation of the source organism. However, the problem is that the products we want to obtain are not always produced at certain conditions. So we would have to modify the conditions, but the problem is that we don't know all the factors which influence the production. So another possibility is to use genetic engineering. We can clone the desired genes responsible for production of the product we want to obtain, and then introduce them into the bacterial cells, which are well known, and we can then control the expression of those genes, leading to production of the large amounts of the products we want to obtain. The problem is that to do this, we need to know everything about the genome of the source organism. So we have to sequence the genome, find the genes which are responsible for production of the substance we want to obtain, and then to extract the genes, clone particular genes into the bacterial cells, and then they control their expression and obtain the large amounts of the pure product, which is not easy but very efficient if you know how to do this.
Now, I will try to clarify two terms that are sometimes misinterpreted: genetic engineering and synthetic biology. Genetic engineering involves artificial manipulation, modification, and recombination of DNA or other nucleic acid molecules in order to modify an organism or population of organisms for discovery or more efficient biosynthesis of products, in our case, marine natural products. Synthetic biology is a field of research in which the main objective is to create fully operational biological systems from the smallest constituent parts possible, biomolecules. The manipulations of nucleic acids are the basics of these two practices. However, it is important to note that genetic engineering alters the existing genetic material, while synthetic biology uses genetic engineering and computational tools to create biological devices, systems, and even organisms that could not have evolved from nature. That's why synthetic biology is sometimes called extreme genetic engineering. In this graph, the 8 stages of transition from naturally occurring organisms to wholly synthetic microbes are presented. In the lowest rank we have natural isolates and naturally occurring microorganisms. Then we subsequently progress through random and targeting changes in DNA. In the end, entire genomes can be created by means of large-scale DNA synthesis. Genetic engineering of less studied, in terms of their genetics, marine species is more challenging than of many known model microbes. That's why heterologous expression of biosynthetic genes, set of genes or whole clusters is more effective strategy for natural product discovery and production. The discovery process comprises 3 crucial steps, starting with gene clusters identification, followed by pathway reconstruction by DNA assembly tools or direct cloning tools, and pathway expression, finally by product detection. In further studies, metabolic pathways may be modified to boost natural products biosynthesis at the industrial scale. Now moving to some examples. Attempts have been already made to produce some of the marine metabolites by genetic engineering of native organisms or heterologous hosts. These metabolites include terpenoids and their derivatives, omega-3 polyunsaturated fatty acids, and compounds classified to marine drugs, originally detected in marine cyanobacteria, actinobacteria, or other microbes. The compound that has already been extensively studied is astaxanthin, carotenoid metabolite naturally produced by many species of microalgae, fungi, and bacteria. It accumulates in higher organisms feeding on the above-mentioned microbes. Due to its strong antioxidant, anti-inflammatory, and anti-cancer activity, astaxanthin is a promising compound for aquaculture, medicine, and nutrition. Since its discovery in 1938, astaxanthin has been obtained at the industrial scale mainly from shrimp wastes, cultures of Haematococcus pluvialis, and Xanthophyllomyrose, or more recently also by chemical synthesis. However, considering the limited productivity of astaxanthin by its natural producers, and the biosafety issues of chemical synthesis where astaxanthin is produced from petrochemical sources, microbial production of astaxanthin via metabolic engineering has become an attractive alternative. Many works have been done on Xanthophyllomyces dendrorose, which has been approved by the FDA for the commercial production of astaxanthin. The carotenoid biosynthesis by overproducing mutant has increased by 24 times in relation to the wild-type yields. Also, by means of heterologous expressions in engineered E. coli, expressing the genes acquired from other astaxanthin producers, higher yields of astaxanthin were obtained. So far, according to published data, best results for increased astaxanthin productivity resulted from combining mutagenesis, metabolic engineering, and improved cultivation strategies. Further enhancement of astaxanthin production will be possible by employing more synthetic biology tools. To sum up, till now, marine natural products production by genetic engineering or synthetic biology has been only pursued at the research level, however with a great potential to be applied at the industrial scale. According to Gerwig and Moore, current challenges and opportunities of bio-based marine natural products discovery and production are in need of new tools to study the cultured marine microorganisms, new expression systems, and awakening of silent pathways for small molecules. Thank you.